Hi everyone, welcome to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. So far, we've held 27 events in this series. We've had so many amazing speakers this year and last year. If you haven't already done so, do check out our archive to watch all of the previous talks in this series. You can find information about each event on our plantae.org blog and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have more events lined up through April. Visit plantae.org/education/plantaepresents to register for them all. Next week, we will be joined by Malcolm and Edith. If you need to leave this webinar early, know that a recording of this webinar will be made available within a few days. Today's talks will each be 20 minutes each. As you listen to each talk, add questions for our panelists in the Zoom question and answer Q&A section. This will make them easier for our moderator to navigate. You'll also notice that there is a chat area open. This section is for you to introduce yourselves, say where you're from, and connect with your colleagues. Today's talk is moderated by Sean Abrams. Sean is a PhD candidate in the Division of Biology at the University of Missouri-Columbia. Their research encompasses phylogenomics, comparative genomics, and metabolic trait innovation. Sean also has a passion for science communication and outreach, serving as a regular contributor to the Molecular Ecologist blog, among many other activities. Thanks so much for joining us today and for moderating the session. I'm going to hand it over to you now.
Of course. I'm really happy to be here and happy to see what great talks we have in store. Just to get started, our first talk will be from Eric Schranz. Eric is currently the professor of biosystematics at Wageningen, see, I even practice it, University. He was an undergraduate at Cornell University and received his PhD from the University of Wisconsin-Madison. He was then a postdoc at Max Planck Institute for Chemical Ecology in Jena, Germany, and at Duke University. Throughout his career, he's been interested and focused on gene and genome duplications and their impact on phenotypic evolution. The title of Eric's talk is Context Matters, Phylogenomic Synteny Analysis of Plants and Animals. Take it away.
Great. Well, thank you, Sean, and thank you, ASPB, for letting us do this. Nice seminar. So hopefully now you guys are seeing my screen. I need to hide that. Does that show up, Sean?
It sure does. It looks great.
Yep.
Okay, great. Thank you so much. Yeah, and as Sean just said, Context Matters will be the title of my talk, and phylogenomic synteny network analysis. And hopefully all of you guys will recognize this as a typical way that we think about synteny. And this is actually a plot where in the middle we have the papaya genome with one genomic region co-aligned to grape VV, one region, to 4 regions in Arabidopsis. So already this gets quite complicated, just looking at one little genomic region where it's been gone through 2 rounds of polyploidy such that you get 4 regions in Arabidopsis, for example. But I'm super excited to be in a moment in science where now we don't just have 3 or 4 genomes like when I started my PhD, but now we have at least 100, if not 200 angiosperm genomes. So remember these colors too, where we look at rosids, asterids, and monocots. I'll use these throughout. So how do we represent synteny across larger phylogenetic regions? And this is an interest of mine. And we've come up with a tool called a synteny network analysis, such that the colors here are represented here, where we can look at, for example, the conservation or genes, for example, here within the Brassicaceae specifically that have been transposed, so unique context. And indeed, I kind of feel like giving a talk, we ourselves, all of us are somewhat transposed into this strange online world to give a talk. But I'll talk to you about genes more. So I think we all know the idea about context mattering, right? It matters what we say, it matters when we say it, it matters how we organize information. So this isn't actually my bookshelf, but it very well could be, right? And there's no reason that we use the Dewey Decimal System. You could also organize your books according to color if you wanted to. And this would be a perfectly reasonable way to think about it. It's the same like when I normally give a talk, I'm standing up giving a talk about context matters in front of people. But of course, we all are dealing with Corona. So here's an example of me teaching to a few students or talking to you. I can't see you all, unfortunately. So I have to believe that you're there. But right, this matters. I will give a very different talk today because I can't see you than if I were standing in front of you, even though I present perhaps the exact same information. And the way we organize information, whether it be genes and genomes or books, there's no right way to do it, right? So this is one of my favorite bookstores in Denmark, this kind of amazing, well-organized bookstore where you can imagine everything is very easy to find. And this is perfectly reasonable. But I might also be able to go to a kind of lefty bookstore to go buy Karl Marx. This may seem like complete chaos, but I'm sure that this woman working here knows exactly where to find me my copy of my book. And right, so maybe I go to one or the other and both work. Similarly, when we think about like success, right, there are different ideas about what's successful. So you might think of somebody like Bill Gates, for example, highly organized, nice and organized desk, everything well set out. And you might think, yeah, that's successful. I want to have a clean desk. But then you have people like artists or Albert Einstein, well known for being messy and kind of chaotic, and actually think that this kind of chaos is important for their creativity and understanding. And I think maybe you see yourself in one or the other of these. Maybe you're a messy desk person, or maybe you're an organized person. And both work, right? And certainly in science, we have many examples of both. And this is one way to think of it, or I also like science fiction and fantasy. So you might also think of like Dr. Spock as being kind of a classic example of highly organized, or somebody who's more chaotic, right? Han Solo, somebody who can just wing it, but is always successful as well. Okay, so certainly when we start thinking about how genomes are organized, you might think of chromosomes, right? And genes within them. And these actually do matter, not just what chromosome they're on, but how they're organized within the genome. And so this is an example of a human chromosome, chromosome 9, with all the different important genes organized along it. And you might think, okay, this doesn't matter so much where a gene is. However, it does actually really matter. For example, you might have out near the tips, like the ovarian cancer gene. And this might make sense that this is where chromosome breaks happen a lot and actually contribute to it. Or you might have some very important genes tucked away in the pericentromere where they're not easily expressed. And indeed, in animal development, you often see very important developmental genes tucked away in highly heterochromatic regions. So they're only expressed for a very limited amount of time during the development. And I think perhaps one of the best ways to understand, or kind of our role model for the organization of genes and their effect on function, are the Hox genes in animals. Such that, for example, in the fly, you have a set of genes that basically are organized in the chromosome in the exact same way that they're expressed during the embryo development, which give rise to the body plan. And you'll see that the fly has one Hox gene cluster, where mammals have 4 due to polyploidy, and this is something we'll come back to, due to genome duplication. And remarkably, they're all in the same order, but these genes are similarly expressed between fly and human, also giving rise to our body shape. And this duplication somewhat gives rise to the increase in complexity, some people think. Okay, so this is kind of a role model for genome organization being recapitulated in rules for organization. But what I like about studying biology as opposed to say physics is while this rule is definitely true across most animals, if you look hard enough, we find exceptions. So here's an example of an animal with 8, all of its Hox genes, but spread across different chromosomes. And you might think, oh, well, how can that possibly be? Will that have implications? And indeed, this is the genome of the octopus. So indeed, many people think that this loss of kind of context has given rise and helped give rise to some of the alternative phenotypes of something like the octopus. So rules can be broken. When context changes, it can have implications. And I think we certainly know this when we think about, I mentioned cancer before. So this is within humans, just examples where you have chromosomal breaks, moving genes or whole chromosome regions around, and you gives rise to fairly catastrophic phenotypes with big problems. Or you might think about when you're making an Arabidopsis transgenic or any sort of transgenic, where you put it in the genome often has implications for its expressed or not expressed, right? I'm sure many of you, this is one of the reasons, for example, CRISPR-Cas is thought to be better. You're just changing the endogenous gene, you're not changing context. Okay, so for a long time, people thought that this didn't happen with plants, that we didn't have this kind of context. But I think one of the most exciting trends over the last couple of years has been the realization that we often have these biosynthetic gene clusters, and that co-expressed genes in, say, trichomes give rise to unique compounds, and these are clustered together. So this is just an example of some of the secondary compounds between potato and tomato, and you'll see there's 2 copies due to polyploidy. These are 2 regions with numerous genes duplicated, many synthetic, many unique to each one, that give rise to the glycoalkaloids. And this is one of the reasons you never want to eat a green tomato or a green French fry, right? If you get a green French fry, you will get sick. You all know that. And these are because of these glycoalkaloid compounds being expressed when there's green in the potato. And this is something I've worked on lately, is the synteny idea, and looking and understanding how some of these biosynthetic gene clusters have come into being. And I already mentioned another big way we know about synteny, especially in plants, is due to genome duplications. We know that if you have an ancestor, you go through duplication, everything should be in 2, but almost immediately we start getting massive gene loss and fractionation such that most things are in single copy. For example, when I talked earlier about one region in papaya matching 4 regions actually in Arabidopsis, for example. Okay, and one thing we can do is take this kind of information and plot it into, example, a phylogenetic tree. Okay, so I mentioned at the beginning, what do? We have some tools if we're just doing several genomes and comparing them, such as chromosome painting of 2 species. We can do things, maybe 8, 12, you start to go a little crazy doing this. But what do we do now that we're in this era of hundreds of genomes? You basically would get synteny hairballs. And this could be a big problem, right? To understand this. So my goal as a biologist has been, how do we deal with hundreds of genomes and make sense of this genomic hairballs? And luckily, hairballs are not something new, right? Everybody, and including us right now, we're on the internet. We're all networked via the web, and such as transportation. We're not using many transportation hubs right now, but I think we all understand the idea of our connectiveness and thinking about networks, that I maybe didn't know most of you beforehand that I gave this talk, but now we're somehow connected. You're listening to me. And we have lots of nice tools to deal with these new sorts of networks. Okay, and so usually we think of networks as being something like a social entity, node being people, and edges being, for example, we are connected because we're online together. So in this talk, what I want to do is instead think about a node being an individual gene, the edge between them being synteny or shared context within a genome. And then if you have interconnectedness, so we can think of this as clades, okay? And maybe I should define synteny for you, but synteny to me means that you have a particular gene and at least 5 of its 20 neighboring genes also show connectiveness or same position between them. So that's just kind of a standard definition of it. Sometimes people think about that being a particular chromosomal context, but in this case, I'm thinking more of microsynteny than macrosynteny, okay? So just to maybe illustrate this, I give you kind of a cartoon version of this where we have several species. So we have species A through G. For example, F and G have gone through polyploidy. So a hexaploid versus, or a tetraploid versus a hexaploid. And most genes are synthetic between them, but some will have different amounts. For example, due to polyploidy or due to tandem duplications can also be put in here. But we might also get something like this that might be a real combined gene or a false one. Okay, so we can represent the synteny this way, but what I want to introduce is this idea of the network, where we pull out this information, and instead of representing it linearly, we represent it as this network. For example, gene 2, where you have basically the genes across the species that have it. As we get more genes, for example, this gene 5, you have many, many more genes due to polyploidy, and also tandems get pulled in, which is quite nice. And imagine then gene 4 and 5, for example, are actually ancient duplicates of each other. They actually can be connected together as well, and we can see that. And that's something I'll come back, for example, like in the Hox genes. And we can even detect these kinds of false, or these joins like this, such that E7 is also connected here. But using a lot of the technology of network science, we can know that this probably is not a good join. It might be an artifact. So we can prune these and eliminate that, or we at least can investigate this when we see not this interconnectedness, but basically one node connected outwards. Okay, so that's just the idea of it. And then we've come up with a pipeline to do this for as many genomes as you want to add into it. So in this case, we take, for example, 100 genomes we did initially, and we compare everything. We detect synteny, we place it into a database, and then we could do this clustering and pruning and look for patterns, such as what I introduced at the beginning. Are things conserved or transposed? Okay, I won't go through the details. If you want to talk to me about it later, I'm happy to. But it's kind of going through this whole pipeline to finally get to what we can understand in terms of phylogenomics, or this interesting patterns of conserved or transposed. Okay, and as a first example, just 4 years ago, we did this on we only had about 40 genomes. That's how many genomes have changed. And we decided to look at perhaps one of the best studied plant gene families, the MADS box, just as an example. And I think everybody, if you're a plant biologist, you know the MADS box controlling floral development, for example, the ABC model with these different functions. What I also really like about the MADS box, though, is you basically have two flavors of them. You have the type 2 that are often these highly developmentally specific ones, and the type 1s that are often involved in, like, say, sexual reproduction. And they have very different dynamics. These tend to transpose and tandem duplicate and move around, and these are highly stable. So as a proof of principle, we could go through this pipeline again of these 41 genomes and do our synteny, and also combine it with phylogenetics. So we did this across these 41 species, and not surprisingly, we were able to identify a lot of clades of well-known MADS box, okay, like Agamis, for example, or Pistillata, or AGL15, for example. But what you can already maybe immediately see is we started to see things like SEP1 with squamosa, or AGL6 with TM3, or SEP3 with SFTs. And this was quite surprising. So why would that be that we would be getting 2 MADS box genes in one cluster? And I want you to think back to that idea of the Hox genes. And indeed, that's exactly what we're seeing, that you tend to have these genes next to each other within a genome. Another way to represent this then is these same colors as synthetic clusters, such that we have many clades that the individual genes form one synteny cluster, like AG is one cluster. But when we have SEP1 and squamosa, you can see they are phylogenetically 2 clades, but are connected syntenically, or AGL6 with TM3. And that's because it's due to an ancient duplication with shared synteny across long distances. Okay, so when we think about this, how can we then go from those patterns to understanding kind of phylogenetic profiling? So how can we find the rules or the exceptions to it? So now we're going to look at those same clusters across then those phylogenetic groups, for example, rosids, asterids, or monocots, and look such that if it's colored in, then everybody has those clusters in them, and we'll already see some jumping ones. So for example, here are clusters that are basically synthetic across all angiosperms. And you can see these, and many of these are these nice duplicated ones, for example, where you have 2, right? So they're connected. But I think what's quite interesting is, where do we see the jumpers? So you might see holes here, and then we see them only in one clade. So for example, in grasses, we see many MADS box genes in unique genomic contexts in green that are not shared with any other angiosperm. Or in the Brassicaceae, we see some very interesting ones, such as FLC, or for example, both Pistillata and AP3, two key B-class genes, are in unique genomic contexts only within Brassicaceae. Okay, and this is just another way to show that for AP3, synteny is highly shared across everybody except Brassicaceae. Similarly with Pistillata, shared across all angiosperms, but in a unique context only in Brassicaceae. So that our model system for floral development is actually the exception. The B-class genes, PI and AP3, are in fact in unique genomic contexts only there, which opens up new ideas for potentially why have they jumped? Why are they in new context? Could they actually have different chromatin dynamics? And if anybody knows anything about Brassicaceae flowers, they actually are highly canalized. You see almost no variation in petal and stamen number, and perhaps this could be partially explained by something like this. Okay, so that was our first attempt to do this with MADS-box genes. We've also done a similar type of analysis with AP2. Maybe some of you are interested in that, and found also basically synteny is highly predictive of this, and we could do this also this phylogenetic profiling across numerous species. But interestingly, we found the jumpers in Brassicaceae again tended to be something that's known as the Leffel genes, and these Leffel genes are involved in, for example, embryo development. So this is something that could be quite interesting for some people to think. AP2s highly conserved across most families, but for example, when it comes to embryo development, unique genomic context of AP2 genes. It's just an example. And we've taken this and also gone into other gene families, for example, the MAM for glucosinolates, also with Sean, the moderator, about this, just as an example. So if you're interested in how we've used this, here's just a few examples in the last year of some papers of gene families we've done. But now I want to end, I think I still have a few minutes to get into this, where I do a big comparison between angiosperms and mammals. Okay, and why would we want to compare angiosperms and mammals? They're phylogenetically quite different, of course, and diverged by hundreds of millions of years. But interestingly, these two clades are actually a pretty good comparison. They both diverged about 170 million years ago. They both are mostly split into three large clades. So for example, you see here the three clades in mammals, the three kind of major clades in angiosperms, for example, the rosette, supraasterids, and monocots that I mentioned before. And you might think that the life history is going to define a lot of these, but if you think about, like, for example, rodents are basically annuals, and over in plants, we also have long-lived species like trees, right? So it's not that funny of a comparison to make. And both have about 100 completed genomes. So pay attention to these colors. So mammals will always be in this reddish color, plants in this bluish color. So what happens when we compare the overall level of synteny between these? And this is just a way to represent any animal genome compared to any other animal genome. And it's basically synteny is very highly conserved, such that almost 80 to 90% of genes between a rodent and humans, or humans and a horse, or humans and a whale is conserved. And this shouldn't come as a surprise. Animal people have it very good that they can identify any gene here. And it doesn't matter, for example, if we're looking at primates, they're still conserved, or primates versus like a cow, they're conserved. What about then when we look at plants? Oh my goodness, the complete opposite, basically, such that we have almost no conserved synteny between, for example, grasses and Brassicaceae, that would be up here. But within a plant family, we have very high levels of synteny. So really, these are completely different beasts. And one of the reasons is clearly then plants just have very dynamic genomes. This should probably not come as a surprise to any of us. And a lot of this is maybe due to the ancient polyploidy. Okay, so one way to think of this is maybe animals are Spock genomes, and plants are Han Solo genomes, basically. Okay, and we can also do then not just look at one gene family like with the MADS box, but we can look at every single gene family. So now here, every single row is a different gene family, and it shows if it's conserved. So if it's across the phylogeny, it's basically conserved, and the color tells me it's in single copy. So basically, almost every single gene is single copy and syntenic across all animals. A few are in duplicate, due to some ancient polyploidy, and very few genes are in unique genomic contexts, such as primate genes in a unique context, or cow genes or rodent genes, for example. Plants, you basically have the exact opposite. Very, very few single copy genes, some high copy genes, such as in more than 3 copies even. You see the color red here, and many, many more of these unique genomic contexts per family. Again, because often you have polyploidy at the origin. So really quite different. So in some ways, the question becomes, what genes then are potentially the special ones? So here, basically 86% of genes in animals are single copy of syntenic, and the unique ones are these 2 syntenic contexts. So who are the rebels here? And just a few examples are, for example, in bats, we have 2 genes involved in DNA repair, for example, that are in unique genomic contexts here and here. And this could be due to flight, for example. These actually were found by positive selection. Some examples from cattle that are unique, or from primates. For example, 2 brain development genes that are unique to primates, unique genomic context. So these give kind of new, interesting hypotheses to be tested. Whereas maybe over in plants, we have something quite different. It's not the jumpers that are quite special, but maybe these single copy genes that are syntenic across everybody. And when we look at this, what's normal in animals, but unique in plants, many of these genes here are actually genes involved in, for example, photosynthesis. And I think this could be quite exciting idea to think about. If you think about a plant, what's the one thing a plant has to do every single day? It has to wake up and fix carbon. Also, for example, the clock has to tick every single day and know what it's doing. And I find it quite exciting and interesting kind of new way to think about plant genomes as potentially these kind of key genes involved in photosynthesis might be the ones that are anchored and really need to be in a particular genomic context. So, and these are the types of things we're thinking about. And I think time-wise, I'm basically maybe just a few minutes over. And with that, I want to say thank you in particular to my former PhD student, Tao Zhao, who did most of this work on this for my funders, such as NVO, to my collaborators. And maybe as a shout out, one of the things that Tao and working also the Van de Peer that we're doing is now taking the synteny network and using it for phylogenetics. So this is maybe something to look out for in the future. And maybe I leave you with this nice quotes, thinking about Einstein. So if a cluttered desk is a sign of a cluttered mind, of what then is an empty desk a sign? So thank you for your time, and I'm happy to hear your questions. So then I think I need to stop sharing.
So great. We already have a question or two, so we can go ahead and get started with that. So we have about 7 minutes for questions. This first question: would it be possible? Would it be possible that the unique syntenic feature of the Brassicaceae is due to a higher number of genomes sequenced as compared to other families, or is it somehow normalized in your analysis?
Yeah, that's a very good question. And indeed, and it could be that we have more power indeed to be able to detect within Brassicaceae and grasses, for example. And that is true, we see the pattern. But basically, as soon as you have 3 or 4 genomes in a family, you're gonna see something pop out. And also genome quality is going to be a big part of it. And the one nice thing about the Brassicaceae is I actually really believe the Brassicaceae ones too, because they're small genomes that assemble well. That used to not be true for a lot of other families, but now I think with long range sequencing, we're getting a lot of really good genomes. So one reason I like this project a lot and this technique is because it's actually something that will get better with more genomes, and will get better with higher quality genomes. And that's kind of where the future is going. So I'm not sure I totally answered the question, but indeed that could be a possibility. But I think the fact that we also see so many jumpers in Solanaceae, we see so many jumpers in legumes, we see so many jumpers in grasses, it's just the reality of it.
Well, then I have a question that might be related to that. So do you see patterns in certain gene families? Like we know certain gene families act differently to duplication. Are there, is it an aspect of just which gene families you are looking at when you are taking a piecemeal approach that you'll see these, or are there overall defined rules of life that you think go?
Yeah, so are you asking a little bit, will certain gene families show different dynamics?
Yeah.
Is that kind of what you're asking?
Mm-hmm.
Yeah. Well, or maybe I'm putting words in your mouth, but okay. So, well, I think that this is one of the interesting things is that, and I didn't point it out in the Mads box one, and I meant to do that, is of course we know that certain gene families have some sort of, yeah, you don't want to over personify genes, but it is true that like disease resistance genes, right? NBLRRs are known to be highly dynamic, form tandem arrays, and move around the genome. And indeed, we see that with our synteny analysis. And things like developmental genes are often these ones that are well constrained and stay put. But even something like disease resistance genes, what I find quite exciting about it is that we do see some of these different patterns appear. That most NLRs are genes that form tandem arrays, everybody in the same order, but every once in a while you find pairs of NLRs that are in opposite orientations, and this stabilizes synteny, and indeed we find these over longer evolutionary distances. So I think even something that I haven't gotten into yet that I think is really exciting is to think about, so certain gene families will have certain dynamics in terms of their conservation or not, but what is the underlying principle behind that? They don't have a mind to say, I'm a jumper, but that means there's something intrinsic in the DNA of that gene that gives it a predisposition to jump or not to jump. So that is the question, I guess.
Mm-hmm. We have I think we have time for one more question that came up in the chat. Are single copy genes more often in conserved syntenic regions?
Not necessarily. Yeah, that's what's interesting. It's not necessarily that's what we think of as single copies. So we actually, in the paper that we did on the overall comparison across big groups, we looked at BUSCO genes. So BUSCO are these kind of single copy holy grail genes that are these conserved single copy ones that people like to look at. And interestingly, we found that many of these BUSCO genes still were in multiple synteny contexts. So I think sometimes our mindset of single copy genes and syntenic genes, we have to be careful in plants. The rules are not nearly as good as they are in animals, and we often come at genomes from an animal-minded perspective. Mm-hmm. So I'm actually skeptical about these single copy genes are often due to some polyploidy, because they all went through a polyploidy, right. And it may be that post-polyploidy it goes back to single copy, but that doesn't mean that copy one is always maintained. It may be copy one in one lineage, copy two in another lineage, right?
Mm-hmm. Very cool. And I think we actually have time to squeeze in this last question here. So the largest syntenic blocks are known to be dominated by evolutionary conserved developmental genes in vertebrates. So animal comparison. This is due to the presence of long-range enhancers. If this is not the situation in plants, could that be inherently because of the difference in transcriptional regulation in general?
Oh, yeah. So this is, I think, whoever said this, absolutely. This is, I think, what's exciting is to think about what are the reasons for this, that we know that genes are bigger, they have more complex promoters, they have all these enhancers in animals, and we have less of that in plants. I think we're getting to these large blocks of chromatin being possible. I don't think I think this is something that will need to be looked at in the future. One thing I would maybe say is I've come from a polyploid background, and I've always thought we see the signature of certain genes maintained in duplicate after polyploidy. And I've always thought, oh, it's a polyploid thing. But now I've started to think, could it actually be that the genes that are maintained after polyploidy are actually because they need to be in conserved synteny, so that we actually see we're not seeing conservation due to polyploidy, but we're seeing conservation of synteny being important. That's not quite the answer what the person is thinking, but it's started to play with my preconceived notions of how genomes evolved after, say, polyploidy? Could it actually be more that we do have conserved development cassettes that need to be maintained in synteny, so they just seem to be the ones that are conserved post-polyploidy, for example, that drive a lot of these plant genomes. So not quite the answer to it, but
no, it's good.
For thought.
You started on polyploidy, and I could keep going for another half hour,
yeah.
but I think it's a good time to transition to Carolyn's talk. So thank you so much again, Eric.
Thank you so much, everybody.
Okay, so our next speaker in this seminar is Carolyn Pont. Carolyn is a PhD engineer at INRAE, Clermont-Ferrand. Man, I should have practiced all of my locations today, in France. She's conducting research on paleogenomics based on sequenced ancient DNA, aiming at deciphering plants' origins and evolution that shaped the present-day genetic diversity of major crops, with a focus on wheat. She's obtained her PhD in 2016 from Clermont-Ferrand University in France and was awarded in 2018 with the MARIE CURIE AgreenSkills EU-funded project postdoctoral fellowship conducted at the Paleogenomics Lab of Beth Shapiro from the University of California, Santa Cruz. Carolyn's talk, Reconstruction of Wheat Origins and Evolutionary Trajectories from Modern and Ancient DNA. Take it away.
Thank you, Sean. Thank you, Eric, also for this interesting talk. So I will share my screen. Is it working?
Yeah, we can see your PowerPoint slide, and then if you go over to the presenter mode, there we go. Perfect.
Perfect.
Perfect. So thank you. Okay. I would like to thank first the organizer for the opportunity to present my work on the reconstruction of wheat origins and evolutionary trajectories from modern and ancient DNA. I work in France at the INRAE Institute in the Paleoevo group directed by Schranz. So wheat is the most consumed cereal in the world and the most cultivated crop after maize and before rice. Wheat is an excellent source of energy, rich in starch and protein, and used for bread and pasta. As you can see here in green, the origin area of wheat and the region is called Fertile Crescent, mainly along the Tiger and Euphrates River in Iraq, Turkey, and Syria. And the Fertile Crescent is it's a limited climb, and right now wheat is cultivated crop worldwide, like in Morocco, Canada, Australia. And the question is how this species with a limited habitat has become one of the most expanded crops. I present here a simplistic model of modern wheat evolution from its ancestor in different colors. At the bottom here, there is the hexaploid wheat, Triticum aestivum, used for bread, and is represented by 3 color cycles because the bread wheat derives from 3 diploid ancestors here at the top after 2 rounds of hybridization. A first hybridization even took place 500,000 years ago between the ancestor of the genome A and B to obtain polyploid species, the tetraploid wide emmer Turgidum dicocoides. And there 10,000 years ago, this wheat evolved into Turgidum dicocum. It was the wheat domestication to obtain the cultivated emmer. And then we also find durum wheat used for pasta. In the same period, 10,000 years ago, a second polyploidization even took place between the tetraploid wheat and the diploid ancestor of the genome D, here in yellow, and to obtain Triticum aestivum spelta and modern bread wheat consumed for bread. We have also modern representatives of the diploid ancestor, A, B, and D. And we will see during my presentation how much this complex is based on recent analysis. There is a big difference between wide emmer wheat and high yield cultivated wheat. Of course, they do not have the same habitat, as you can see on the picture in Israel, but also the wide emmer wheat presents a major desarticulating spike. Brittle washy species have been domesticated to have a non-brittle spike, like in durum wheat or bread wheat. And the question is how this species has become one of the most expanded crops. Of course, in 2021, there are very efficient production crop system. But before that, certain genes have played a major role in the domestication. 3 genes are clearly described. The most important domesticated genes is the Q gene, where is the Q gene where a spontaneous mutation affects spike shape and give the free threshing. Here in red, the square head notice QQ in red. And the small Q allele in white wheat confers splatoid spike phenotype characterized by a lax head and with elongated rachis and non-free threshing seed. Secondly, here in green, there is a brittle washes genes. All the white species are the natural seed dispersal mechanism. And thirdly, here in blue, the emmer wheat has genes that confer tenacious gloom called whole grain that underwent genetic mutation to give rise to fully domesticated free threshing bread wheat. So as you can see here in red, durum wheat and bread wheat have a naked grain and non-brittle rachis. So it was the introduction, and now we can speak about paleogenomic, or how in my group study with origin to better understand the evolutionary history of this complex genome. And we use two approach. First one is based on synteny and genome reconstruction from modern genomic data called synchronic approach. Here we will see how we use this the exome sequencing for tracing the ancestry of genome of modern bread wheats. And the second approach on the right part is based on ancient DNA sequencing from wheat archaeobotanical remains. And this method is an allochromic approach to reconstruct the plant evolutionary trajectories by comparing the ancient DNA. So first one, the synchronic approach. And to better understand wheat evolutionary history, we have investigated SNPs dynamic in wheat in the frame of the European Will Be project. It was a large consortium with 18 partners led by Gilles Charmet from INRAE to combine genomics, genetics, and agronomy approach to improve the European Will Be and barley production. My work in my group was to focus on ancestry of modern bread wheat. So here we study variant diversity in wheat using a large panel of 487 wheat accession covering 68 countries. In this panel, we have a few white species, 27, and a majority of hexaploid accession, 435. This hexaploid panel has been divided in 4 groups of land races for older accession before 1935, all cultivar between 1936 and 1965, cultivars before 1985, and then modern varieties after 1986. As you can see on the right part, we have a huge diversity in this panel with few diploids, but also different kind of polyploids wheat. In this project, exome capture and sequencing was performed for each accession with Nybergene and at the Earlham Institute. More than 600,000 variants of SNPs and Handel were identified on the panel, covering more than 41,000 genes. From this data, we investigated the SNPs on the 3 subgenome A, B, and D across more than a century of breeding. For that, we compare landraces, cultivars, and modern varieties using the diversity index TAGIMAS-P. And as you can see on the right part, for each genome, we note a temporal erosion of diversity with the highest diversity in the oldest group, the landraces, and lowest in the most modern group of genotypes in orange. This reduction on diversity is asymmetric between the subgenome with more diversity in the A and B genome compared to the D. And the D genome is the less polymorphic genome with a 6% variation. Using a sliding window approach to deciphering local reduction in diversity and taking into account the geographical structure between wide-width ancestor and hexaploid landraces, we have identified 1,200 regions contrasted between wide-width ancestor and hexaploid landraces and support a huge heterogeneity in the reduction of diversity during the domestication process along the wheat genome. As you can see on the picture, these regions are distributed in clusters across the genome. And domestication genes conferring beta rachis, tenacious bloom, and non-free tracing characters, the Q gene, were identified in close proximity. And secondly, we identify more than 5,000 polymorphic sites with breeding signature based on the individual centric method PCADAPT between groups. No genes, including genes for photoperiodism, vernalization, reduced age, as well unical genes driving plant architecture were located close to this improvement signal. We have noticed in our data that a lot of polymorphism are shared between tetraploid wheat and hexaploid wheat, which is not expected considering the hexaploid species appeared 10,000 years ago after a rare polyploidization event. And in our panel between the two species, we have more than 335,000 shared SNPs, corresponding to 56% of initial number of SNPs. And this phenomenon can be explained by a recurrent hybridization between hexaploid and tetraploid wheat. Of course, these shared mutations are carried by the A and B genome, inducing de facto a reduced diversity in the D genome. The study of network phylogenetic inference of 487 wheat accession has shown a reticulated evolutionary scenario. Here we can see minimum spanning tree of the phylogenetic network, maximizing the weight of cap age. And the resulting graph highlights the vertical evolutionary signal linking diploid and tetraploid and hexaploid wheats. And you can see that for bred wheat, there are 3 independent clusters, and yellow, red, and blue. And these clusters correspond to the geographical structure, as you can see on the right part in the world map, where is presented the country of origins of the 3 independent clusters, with the color shading illuminating the number of genotypes. So we observe a reticulated evolution of Triticum aestivum, then leading to additional younger wheat communities. In red, the Western Europe line originating 1986 or later, mostly comprising winter wheat, cultivar, and current varieties. And in blue, Eastern Europe lines originating from countries formerly part of the Warsaw Pact. So these 2 pools of varieties are genetically different. To summarize, we've shown this a complex origin of wheat genome we've called a reticulated evolution with hybridization or gene flow between Triticum durum, Edicocum, and aestivum, explaining observed contrast in diversity between subgenome A, B, and D. So now we will see the study with origins in allochronic approach with ancient DNA from with archaeobotanical remains. So since the Neanderthal DNA sequencing in 2006 and the Siberian woolly mammoth in 2008, a lot of study begin to be done in plants with several species, barley, grape, maize, and different tissue, grain rashes, and up to 10,000 years old samples. So the ancient DNA story requires a very particular approach, very accurate, and which allow the authentication of the sequenced samples. So the precious samples are collected and identified by archaeobotanists, and the quality here depends on the age of the samples, the conservation also. It could be desiccated, waterlogged, or shared, and the type of tissue. The grinding and the DNA extraction, as you can see in the picture, take place in an overpressure confined lab to avoid environmental and human contamination. And after extraction, the DNA is very rare and degraded with sometimes inhibitors, but it's still possible to prepare a DNA library for sequencing. And at this step, it's possible to choose two different ways, a shotgun approach or a gene capture to target a specific region or a specific species. And the objective here is to obtain a high level of endogenous and unique reads. Then our work is to map the sequencing reads, but also to complete authentication. For that, we perform the damage pattern, as you can see in the yellow box, with the map damage tool. The DNA has a specific molecular feature of ancient DNA, in particular deaminated cytosine at the end of short fragments. And another important step is to characterize the level of contamination of the samples by environmental DNA, soil, water, or human. As you can see in the gray box, the taxonomy analysis and microbial profiling could be done with tools like Megan, for example. And only at the end, genetic analysis could be done with demographic history, phylogeny, selection signature, and so. So in my group, we focus on agriculture spread from the Fertile Crescent, as you can see on this map. And expansion of farming in Western Eurasia took more than 5,000 years across different roads. And ancient DNA can inform us about how agriculture spread across the European continent and how hunter-gatherer communities progressively transformed into sedentary farmers. What were the characteristics of ancient crops in relation to modern species? Where took place hybridization between weeds, for example? What are the natural markers for crop biodiversity that have been selected and conserved, or in contrast, lost over the past 11 millennia? So in this topic, we have three main projects. One payload case study with two sites, 4 samples, 5,000 years old. And then an INRAE project for 4 years with 65 samples, 55 sites, and 88 samples, 10,000 years old. And thirdly, a local project study Alpine Lake with 11 excavation on protected UNESCO site on a lakeside village. I will speak with a few results only for the payload case study. And this payload case permit to validate the proof of concept for sequencing waterlogged remains from lakeside village. Because under normal condition, non-carbonized plants decompose rapidly in the soil, and the low rate of oxygen present in wetland considerably slow down this decomposition process. So we will see that it's possible to sequence waterlogged samples 5,000 years old. So the sample presented here was identified by Caroline Charles 15 years ago in the French UNESCO site Chalin 19, lake in Jura. And the method presented was used to sequence these samples, and the bioinformatic analysis gave us more than 70 million raw reads and 70,000 reads mapped on the reference genome, with a mean length of 53 base pairs, so really short fragments, and 92% of unique reads. Here we can see the damage profile with an increased mutation level at the end of each fragment. And on the right part, we can see the taxonomy analysis. And here we show the proportion of reads sequence found in the samples. Most of them come from microbial contamination, little of human contamination here in red, and the interesting part in blue of wheat sequence. Here on the tree, we can find all plant species sequence in the sample, with most of them are Triticum aestivum. This work was done in collaboration with Eva Maria Glegg and Thierry Cange to perform the gene extraction protocol, and also Beth Shapiro in a paleogenomic lab in Santa Cruz. Here I show some mapping and SNPs identification of the ancient DNA. Reads are presented here in several colors, depending on the direction of the sequence, but we can see mismatches. And the most important job here is to separate mutation with low coverage or sequencing error, and SNPs with good coverage, like in green. And for the samples presented here, if I count mapping reads on each ABD subgenome and Triticum aestivum reference genome, I found only reads on the A and B subgenome. So this sample seems to be a tetraploid wheat. But as we have seen, there are several tetraploid wheats, and only 2 have been completely sequenced. The wild species here in orange, and the durum wheat used for pasta here in gray. And as you can see on the right part, I have less mismatches with the tetraploid durum wheat than the hexaploid or the wild species. To summarize, our sample seems to be an ancient tetraploid wheat, close to Triticum dicoccum. But for these samples with a low sequencing coverage, I got only few SNPs, 348 share with the will-be panel, because most of the panel are red wheat, hexaploid wheats. So now I need to perform the analysis with modern tetraploid wheat panel and sequence capture for domestication genes, for example. So to conclude, we identify more than 600,000 variants covering 41,000 genes. We have seen an asymmetric reduction of diversity during the last century of breeding. We have identified also breeding signature, more than 5,000, and domestication signature, more than 1,200. And we have seen that ancient DNA, with ancient DNA, it's possible to identify ancient variants to characterize loss of diversity at key gene, for example. And the main message of my talk is that modern wheat derived from a reticulated evolution between diploid, tetraploid, and hexaploid wheat ancestors, and from the Fertile Crescent 10,000 years ago. I would like to finish for that, and thank you for attention. And I would like to thank all the people working with me on this topic, of course, the director, Jerome Sals, and all the bioinformatic guy, Vandril and David, all the partner on the will be project and will be a consortium, and the collaborators for ancient DNA project with Manon Cabanis and Caroline Schall, they are archaeobotanists, and Beth Shapiro and Anne Green from the UCSC paleogenomics lab. Thank you.
Thank you so much, Carolyn. We have about 6 minutes for some questions. And please, if you have to go, but you want to leave a question quick, we can answer it and you can check back for the recording to see how it was answered. Okay. So while we're waiting for some people to type up their questions, I have two. First, I was wondering, you said there was a reduction in diversity in more recent breeding efforts for wheat. Do you think that is because of because of how we're breeding, because of novel breeding practices, and is just not as important to include the diversity? Maybe not important is the wrong word, not as common to want to include diversity in how we're planning these out, when before it was a little bit more random? Or do you think that's just an age thing? Because they're more recent, they're not going to have as much diversity.
Yes, that is a good, this is a good question, because yes, of course, after a century of breeding, of course, the human process select only the high level wheat with high productivity yield. So of course there is this create a really loss of diversity. We have seen this with between land races and between recent cultivars. So we have really a loss of diversity due to the human selection. And also there is another part that is a reduction from the Fertile Crescent. So most of the diversity is present, were present in the Fertile Crescent 10,000 years ago and the last. So during the expansion, during the road expansion, we lost we lose a lot of biodiversity, because there is some adaptation, there is some yeah, because there are different kind of climate, environmental climate, and also different pathogen, but each time we can create a loss of diversity for that. Yeah, since 10,000 years ago.
Right.
It's observed in lots of species, in fact, a lot of, even in human process. Yeah.
Mm-hmm. And I think Eric may have a follow-up question to that. Can you see any signatures of mutational breeding by European or Eastern European USSR breeders? So do you see?
Yes, there are. In fact, the biodiversity of the panel that we study, we have really a group, a cluster of European weeds that they are really specific allele of specific genes, and so they are really genetically different from the. We have seen there are 3 main clusters, and they are really genetically different due to this human selection, human breeding.
Very cool. Yes, and my my follow-up question with the time that we have left here. So on your presentation on page 13, you showed a network. You said it was like a phylogenetic network. What were the edges? Is that most common? So the nodes were individuals, I assumed, but then were the connections in that network, were they just the most common relative, or was it everyone in a specific clade? Do you know how that figure was made?
Yes, I understand which figure you are talking about, but you asked me if there are several each point in this figure, each point it's a phenotype. So depending of the allele of there are 41,000 genes. So depending of each allele, we can obtain clusters. And so in this cluster, we can see diploids, we can see ancestors, and we can see allele and genes shared between, for example, Triticum durum and Triticum aestivum. And then the other communities or other clusters. So this is a profiling for C hybridization or reticulated evolution.
All right, well, with that, are there any last questions? We have about maybe a minute left. No? Oh,
It looked like there was one, Sean.
Yeah, I see. Hold on. Okay, so last question. I wonder, can we determine the methods how the wheat spread across the world based on genomic data? Was it by humans, or were there other animals, birds, etc., factors that contributed to the spread of wheat across the world other than just humans trading?
The question is that the spread of agriculture, of domestication.
Mm-hmm. Was the spread of wheat across the world, was it largely human influenced, or were there other natural processes that were doing that? And can we see that in the genomic data?
Yeah, largely human, because the road of wheat expansion is really the same when it's close to the Neolithic process, the Neolithic road. So there are the Neolithic from the Mediterranean road and from the Balkan also. So they are really the same. They are main road for lots of species.
Okay. All right. Well, thank you all for coming. I'll pass it back over to Katie.
Awesome. Yes, thank you, Sean, for moderating, and thank you to both of our speakers. Thank you again for all of our attendees that are here today, and for all of you that are listening to this as a recording. It's great to all be here and be able to connect. We hope to see you at our next webinars, and remember to join ASPB. Bye, everyone.
Thanks.
All right.